All right, before we get started on microbiodiversity, assignments so far have been the Macrobiodiversity Lab, Unit 3 Reflective Writing, and Homework 1. I think all of you have looked in both, and you've completed both of these first two. I haven't looked at Homework 1 yet, but if you haven't done any of these, please do that as soon as possible. And for upcoming assignments, today we have the Microbiodiversity Lab. Homework 2 will be posted today and will be due on this Friday. And then homework 3 will be another key terms list. And so that'll be posted later this week, and that'll be due next Tuesday, the 28th. So for today's lecture, we're going to be talking microbiodiversity, and we're also going to be doing a microbiodiversity lab assignment, pretty simple. It's just kind of looking up microbes and recording their characteristics. So when we talk microbiodiversity, going back to the tree of life with the 3 domains of life, microbiodiversity is actually present in all 3 domains. So we're talking about microbes here. And so, of course, bacteria and archaea are all microbes. So all the diversity in bacteria and archaea is microbiodiversity. And then part of the eukaryotes are microbes as well, and those are known as protists, and we'll discuss that a little more. So microbiodiversity is probably more commonly referred to as microbial diversity, and this is, of course, the diversity of microbial organisms, and these are present, again, in all 3 domains of life. So bacteria and archaea are all microbes, and then some eukaryotes are microbes that are known as protists. And today, some of the things we'll talk about are what are the defining characteristics of the different types of microbes, where do these microbes live, and what do they use for energy. And one thing that I definitely hope that you take away from this lecture is that microbial diversity is very vast. They live pretty much anywhere, and they do a whole lot of really interesting things. So in terms of characterizing microbes, one of the challenges that we face is that up to 99% of the microbes in certain environments cannot be cultured in the laboratory. So most of the bacteria and other microbes on Earth, you can't necessarily just pick up and grow on an agar plate or in media or anything like that, because they'll only grow in very certain environments. So initially, prokaryotes were considered to be one kingdom, and their characterization was based mostly on their biochemistry, morphology, and metabolism. But then later, as sequencing technologies advanced, a new classification came into acceptance, which is the 3-domain system that we now know about, which includes bacteria, archaea, and eukaryotes. So we'll talk a little more about why bacteria and archaea were divided from one another and the microbial life within the domain eukarya. And so today we know of a lot more microbes than we ever have been able to in the past because of some of these next generation sequencing technologies. So, for example, what we can do today is just take a sample of like ocean water, for example, and deep sequence just kind of everything in there to detect the basically diversity of microbes within that sample. And so again, because up to 99% of these microbes can't be cultured, this is something that we never really had the ability to do before these sequencing technologies. And so it's only very recently that we've been able to kind of get a better grasp on just how diverse microbial life is. So looking at just kind of the structure of life again, so we have prokaryotes and eukaryotes. And so we talked previously about some of the defining features of eukaryotic cells, and then prokaryotic cells have some differences, mainly in that they lack certain things that eukaryotic cells have. And so prokaryotes include bacteria and archaea, whereas eukaryotes are of course, are all of domain eukarya. And so just as a reminder, while this tree makes it look like bacteria and archaea are more closely related and then eukaryotes are more distant, in terms of last common ancestors, actually eukarya and archaea share a common ancestor more recently than archaea and bacteria do. And so evolutionarily, archaea and bacteria are actually a bit further apart than archaea and eukarya, which is very interesting. But we still characterize archaea and bacteria as prokaryotes. And in terms of defining features of prokaryotes and eukaryotes, just some very simple ones. Prokaryotic cells are relatively small and more primitive. And I don't necessarily like the word primitive to describe bacteria because it makes them sound perhaps poorly adapted, which is not the case. But what we mean by primitive is more that they may, in terms of their cellular structure and their genetic sequence, they may more closely resemble the last common ancestor than eukaryotic life does. And prokaryotic cells also lack membrane-bound organelles. And so these, of course, again, include the domain archaea and the domain bacteria. And then eukaryotic cells, which we've already talked about, are cells that are relatively large. And so to give you an example of, kind of the size difference between eukaryotic cells and prokaryotic cells, in a human cell, for example, basically any cell in the human body, you could probably fit hundreds or even thousands of bacteria. And so eukaryotic cells are that much larger than prokaryotic cells. And they're also more complex. As we learned, they contain membrane-bound organelles, as well as a nucleus and mitochondria, etc. And so we'll recap quickly first on the domain eukarya. So again, characteristics that all eukaryotes share include a plasma membrane or a cell membrane, which is different from a cell wall. And so some eukaryotes do have a cell wall, but not all of them do. Eukaryotes have a cytoskeleton, which is composed of microtubules and filaments. They have membrane-bound organelles like the Golgi apparatus, the endoplasmic reticulum, and the mitochondria. Of course, they have mitochondria, which all prokaryotes lack. And they also have a nucleus with linear chromosomes. And so bacteria and archaea actually tend to have circular chromosomes. So we've learned about how our chromosomes wrap up around histones and form these interesting structures, especially during cell division. But they could also be stretched out kind of end to end in a linear fashion, which is not the case with bacterial chromosomes. So this is, again, is a view of what the eukaryotic cell looks like. So again, nucleus, DNA within that nucleus. We have the Golgi apparatus, these membrane-bound organelles, mitochondria, etc. The cytoskeleton, which you can see really clearly over here on the right. And then one thing that I want to point out is that the eukaryotic cell has what are called ADS ribosomes. And so this is basically the molecular weight of the ribosome is a certain structure. And one interesting thing is that when you look in the mitochondria, they actually have their own ribosomes, which is a 70S ribosome. So I'll talk a little about this in terms of the bacteria and archaea, but bacteria and archaea both also have 70S ribosomes. And so this is more evidence, actually, the fact that mitochondria have their own ribosome, which is distinct from that of our cells, that they are actually just an ancient, basically free-living organism that was came into this symbiotic relationship with eukaryotic cells. And so basically all living things have their own ribosomes, whereas things like viruses do not. So viruses don't encode ribosomes. So ribosomes, or the translation machinery, are actually one of the things that we kind of use to separate life from what we consider non-living organisms. So microbial eukaryotes are called the protists. So Protista was once considered a separate kingdom, but it's not really commonly thought of that now. And the reason for that is that protists contain microbial eukaryotes that fit into, that basically closely resemble animals, plants, or fungi. And so while we consider them one group because they're all microbes, they actually don't resemble one another as closely as they do organisms within either animals, plants, or fungi. So the protist is not necessarily a kingdom, it's just kind of this separate grouping of all the microbes within eukarya. So all protists share the characteristics, of course, of all eukaryotic cells, and they're considered, again, more complex than prokaryotes. So most of these are single-celled, but some also form into colonies or filaments. Protists are diverse in size, shape, and lifestyle. So they include both heterotrophs and autotrophs, like photosynthetic algae, for example. And Protista also includes pathogens. So things like Plasmodium, which are malaria parasites, as well as Giardia and Toxoplasma that maybe you've heard of. and then also filarial nematodes. So if you've ever heard of the disease that manifests as elephantiasis, these filarial nematodes are actually the cause of that disease. So in terms of their diversity in morphology and lifestyle, so you can see just a little bit of the really intricate morphological structures that these protists can have on this slide. And so I think most of these are probably algal species. And yet you can see they have very diverse outward appearance in terms of both size and shape. And then in terms of the kind of complexity of these microbes, so this I'm showing you is the Plasmodium life cycle, or basically the life cycle that a malaria parasite will undergo from mosquitoes to humans. So in humans, initially when humans are bitten by a mosquito, that bite will inject malaria sporozoites. And so this is the initial life cycle of the parasite. These will then enter the liver and go through these various life cycle stages where they take different, basically morphological and other forms throughout their life cycle in the human body, and then different forms even yet once they're back in the mosquito. So again, Plasmodium, while it's just a single-celled, seemingly simple organism, it has this very complex life cycle. And in each of these different stages, of course, it's going to be undergoing gene expression changes, and a lot of different, basically has to adapt to a lot of different environments, because not only is it living in different organisms, but it also has to deal with their different immune systems. So that's an example of one very interesting eukaryotic microbe. So now we're going to move into the bacterial domain, which of course are prokaryotes. And so characteristics that all bacteria share include the fact that they are unicellular, of course. They're ubiquitous in nature, which means that they grow pretty much everywhere. Bacteria lack membrane-bound organelles, and they also lack a nucleus, but they do have what's called a nucleoid, and so this is not membrane-bound. So their DNA is a little bit separate from the cytoplasm, but it's not separated by an actual membrane-bound nucleus. Their DNA content then, as we talked about, they have mostly circular chromosomes. And so rather than having linear chromosomes that wrap up into chromosomes like ours, bacterial chromosomes are actually just an enclosed circle. And this circle will kind of wrap and coil around itself. So it'll end up looking, very kind of coiled up, sort of like ours, but at the end of the day, the genome is enclosed, and so it's just this this circle itself wrapping into these structures. Bacteria also have a cell wall that contains peptidoglycan, which is something that makes them that separates them from archaea. So when we look at the bacterial cell, what we can see is that, again, we talk about this nucleoid that contains their circular DNA, and so it's wrapped into these structures, but it's still a circle. Bacteria also tend to have, oftentimes they have plasmids, which is this extra chromosomal DNA that can encode separate functions that's not normally in their genome. And so we've talked a little in the past about horizontal gene transfer. And so bacteria can also just kind of swap these plasmids in and out between each other. And so that's a really quick way that they can basically undergo evolution and gain these new alleles or functions based on genetic content that's not even included within their genome. And then bacteria can take a number of different, shapes and morphologies, including these, coccus structures, which are just kind of circles. And then sometimes, while they're still single-celled organisms, they can group up into these little colony structures. You also have these rod-like structures as well as spiral structures. And so those are illustrated again here. You can kind of look through if you'd like, but these are kind of just some of the various bacterial morphologies that we know of. And so one thing I want to highlight again is that bacteria are extremely diverse. And so this is that sort of new tree of life that we talked about in the last lecture, where everything at the top here are bacteria, and all of the archaea are contained right here, and eukaryotes are only right here. And so bacteria is clearly the most diverse domain of life. And so there are 92 bacterial phyla, while there are only 26 archaeal phyla and only 5 eukaryotic supergroups. So again, bacterial diversification is a major feature of this tree of life. And the reason for that is that bacteria have very different lifestyles and very different metabolisms. So what I mean by that is that bacteria basically have the most diverse metabolism of any of the domains. And so they include basically every nutritional type that we know of. So photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs or heterotrophs. So when we talk about eukaryotes, we're mostly talking about simply heterotrophs or photoautotrophs. So some of these nutrition types are actually exclusive to bacteria. So photoautotrophs, for example, these are bacteria that kind of function like plants in terms of their nutrition. So they get their energy from sunlight, and their carbon source is CO2 fixation. So they take CO2 out of the atmosphere or whatever environment they're in, and they use it as their carbon source. And so this includes cyanobacteria and purple and green sulfur bacteria. Then there are also photoheterotrophs. And so these are different from plants in that while they get their energy from light, they're actually consuming organic compounds. So just like we eat sugars, fats, and amino acids, so do these bacteria. So this includes some purple and green bacteria. Then there are chemoautotrophs, which actually use inorganic compounds as an energy source. And so this is really interesting. They use things like hydrogen sulfide, nitrogen oxide, or just hydrogen gas actually as their energy source. They can also use CO2 fixation for their carbon source. And so this includes some bacteria, but actually many archaea are chemoautotrophs. And then there are chemoheterotrophs or heterotrophs, which this is similar to our kind of lifestyle, in which their energy source comes from organic compounds, so does their carbon. And so this includes most bacteria and then also some archaea. So in terms of bacterial diversity, bacteria are actually the most abundant life form on Earth. And this one bacteria in particular is probably the most abundant organism on Earth. This is called Pelagibacter ubique. And again, it may be the most abundant organism on Earth. And so up to 1/3 of all of the single-celled organisms in the ocean are actually this bacteria, which you can imagine the microbial diversity in the ocean is very vast. And for this to take up 1/3 of that is just very remarkable. And so because there's so much of this bacteria, it actually has a major role in the Earth's carbon cycle, just because so much of Earth's carbon is actually present in this bacteria. So bacteria also play crucial roles in all of the nutrient cycles. So soil bacteria, for example, are crucial in the nitrogen cycle. They're methanotrophic bacteria that break down methane, as well as decomposing bacteria and etc. And so in humans, some bacteria can cause disease, but the vast majority are beneficial, and we've talked about this, and even necessary for our development and our health. And so we have more bacterial cells actually on and in our bodies than we have our own cells, which is fascinating. And it's, we're capable of having that because of how small bacteria can be. And so, of course, our gut microbes are very crucial to our health. And if you ever search microbiome and health or anything like that, you'll find a number of things that people think that the microbiome contributes to today, including even things like mental health and the early brain development and things of that nature. So bacteria are absolutely crucial for our lives as well. So one bacteria I wanted to highlight, mostly because I used to study it and I think it's really fascinating, is Wolbachia. And so Wolbachia is the male-killing, gender-bending, and gonad-eating bacteria. So this is a gram-negative obligate intracellular bacteria, which means that it has to grow within another organism's cells. And so Wolbachia is very common in arthropod species, and in fact it's present in up to 65% of all insects. And the way that Wolbachia is transmitted is vertical transmission from the mother to the offspring. So it's transovarial transmission. And so in order to ensure its spread, it can actually have a number of interesting phenotypic effects on its hosts. And so these include cytoplasmic incompatibility, feminization, and male killing. So why would it want to do these things? It's because it's transmitted from mother to offspring. And so Wolbachia is not transmitted from males to offspring, and so it has a number of ways to basically give reproductive advantages to females that are infected with, or just turn males into females that are infected with it. And so these, of course, are evolutionary measures to ensure its success and spread through populations. And so Wolbachia may be the most successful endosymbiotic bacteria that exists in still a free-living state. And so cytoplasmic incompatibility I wanted to highlight. So basically what this means is that, so of course when you have a female who's uninfected and a male who's uninfected, they'll be able to produce uninfected offspring. Now, when Wolbachia infected males come into the picture, they actually cannot mate successfully with uninfected females. So the offspring of this cross will actually die, and that's called cytoplasmic incompatibility. And what this does is that it actually gives a reproductive advantage to infected females, because an infected female can mate successfully both with an infected male or an uninfected male and produce infected offspring. So you can see that the infected female in this situation, assuming an unequal balance of infected and uninfected males, is actually twice as reproductively successful. So this is one mechanism that Wolbachia uses to ensure its spread through populations. So I wanted to highlight this because, while we talk about bacteria are considered primitive and things like that, they actually have evolved numerous fascinating mechanisms to ensure their success in extremely diverse environments. And again, the fact that bacteria are the most abundant organism on Earth really highlights the evolutionary success that they've had. Okay, so let's move on to the domain Archaea. So characteristics that all Archaea share is that they're unicellular. They're not known to express virulence genes, and so Archaea do not include pathogens that we know of. So this is one way that they differ from bacteria. Like bacteria, they lack a nucleus but have a nucleoid that's not membrane bound. As far as we know so far, all Archaea have circular chromosomes. They also have plasmids like bacteria. So the archaeal cell wall actually lacks peptidoglycan. So while the bacterial wall is always composed of it, Archaea do not have peptidoglycan. So that's another way that they differ from bacteria. They're also insensitive to antibiotics, and the reason for this is that, for one thing, they have a different cell wall structure, so for reasons like they don't have peptidoglycan. So a lot of antibiotics target the cell wall. So since theirs is different, they're insensitive to antibiotics. And then some antibiotics also target the bacterial ribosome, but Archaea actually have ribosomes that are more similar to eukaryotes than bacteria. However, their ribosomes are still distinct from both. So these are a few things, a few really good ways to kind of distinguish Archaea from bacteria. So speaking of archaeal diversity, so Archaea include many extremophiles that can live in environments with like extreme heat or salt or other extreme conditions. And so Archaea were initially discovered in these geothermal volcanic vents. And so it's really interesting that they can survive in that environment. So Archaea are predicted to account for around 20% of prokaryotic life in the ocean, as well as 1 to 5% of that in soil. And then they're probably the most common life form in marine subsurface sediment and geothermal habitats. So bacteria, upon their initial discovery, were actually thought to only include extremophiles. But we now know that that's not the case. They're actually pretty ubiquitous in nature as well. Not quite as abundant as bacteria, but there are a lot of Archaea out there in a lot of different places. And so the reason that Archaea became their own kingdom is because while they look and kind of act in many ways a lot like bacteria, once our sequencing technologies kind of progressed, we found that some of their genetic content, especially like their ribosomal RNA, is actually more similar to eukaryotes. And so on that tree of life, again, it's not kind of this one last common ancestor that branches off in 3 different directions. Archaea and bacteria basically split first, and then Archaea share a common ancestor with eukarya more recently than with bacteria. So Archaea and humans, these are part of our microbiota as well, and so these are actually mutualistic. So, for example, they are methanogens in our gut that help us to break down food. And archaea have very diverse metabolism like bacteria. So archaea do a lot of kind of interesting things and use interesting things as energy sources. So they can use, like us or like most bacteria, organic compounds as an energy source, but they also use weird things like ammonia, metal ions, and even hydrogen gas to get their nutrition. So like bacteria, archaea are crucial for nutrient cycling, especially the carbon cycle. And so archaea are actually the only organisms known to undergo this methanogenesis process, which is the formation of methane by microbes. So this is the final step in the decay of organic matter. So carbon-based compounds, as they decay, ultimately they will form methane. And so again, this process is only performed by archaea, and it's absolutely crucial to carbon cycling and basically the recycling of carbon so that it can be used from the soil by plants and basically cycled again, through the food chain. So archaea are obviously crucial for that reason. When we look at the archaeal cell, they look quite a bit like bacteria, and this is why they were initially, classified in the same kingdom as bacteria. And so they have, again, the cell wall like bacteria. It's different from bacteria, but looks pretty similar. They can have flagella or pili like bacteria. They have their own ribosomes, and so bacteria and archaea both have what's called the 70S ribosome. However, the archaeal 70S ribosome is actually more similar to the eukaryotic 80S ribosome than it is to the bacterial 70S ribosome. So that's more evidence that we share a more common last ancestor. So while archaea do look pretty similar to bacteria in morphology, some have unique morphologies, such as these square shapes, as well as the branched shapes or lobed structures. And so archaea are actually a bit more morphologically diverse than bacteria, despite the fact that bacteria are more metabolically diverse than archaea. And so that does it pretty much for the 3 domains of life and kind of what I wanted to cover in terms of microbial diversity. And so with this, go ahead and fill out the microbiodiversity lab. So the lab report basically just has a table with different microbes, and it used to be that they had microscopic specimens to look at, but since we can't, use the microscope, we're just going to look them up on Google or whatever and take a look at their pictures to kind of get an idea of some of the microbial diversity that's out there, and then record the appropriate data in that table and submit it on Moodle. And we will come back Thursday and talk a little more, kind of going back to biodiversity in general. And then next Tuesday, we'll have no class, and then the quiz will be a week from this Thursday. So I'll see you guys Thursday.